Hi everyone, I am Li Shuangying, and I'm from South China Normal University, Guangdong, China. It is my great honor to be here to introduce our work on diffusion model for RNA sequencing data synthesis. Since the reason of visa, I cannot present it in person here and apologize that. Okay, anyway, let's begin. The paper title is Probabilistic Flow Diffusion Model for Accelerated High-Quality Single-Cell RNA Sequencing Data Synthesis. And as we all know, biological systems are extremely complex and driven by nonlinear interactions of thousands of molecules within and between cells that are crucial for understanding physiological and pathological processes that are the key to developing new therapies. However, single-cell RNA sequencing faces a challenge: limited sample sizes due to financial, ethical, or patient availability constraints. Small datasets may not fully capture biological diversity, leading to unreliable results. While some projects, like the Human Cell Atlas, offer the broad insights, they lack specificity for rare cell populations. This makes generating high-quality, controllable virtual cell data a critical topic in current computational genomics. So that is why we do this research, and it is sort of the part of AI for science. And the existing methods for generating single-cell RNA sequencing, we have like the statistical methods GAN, VAE, and diffusion. Statistical methods usually involve clustering, dimensionality reduction, and regression. They can capture the marginal distribution of read counts to a certain extent, but it is difficult to learn the joint distribution of these counts. To address these limitations, generative models such as VAE and GAN are used to synthesize new single-cell RNA sequencing data by learning from existing datasets. Although the SCGAN pioneered the use of GAN in computational biology, the quality of its synthesized data is average. However, these models still cannot fully address the complexity of RNA sequence data synthesis. Compared with VAE and GAN, diffusion models have better sampling efficiency and are less prone to mode collapse. Also, someone introduces the transformer architecture and uses the denoising diffusion implicit model for accelerated sampling. While it accelerates the generation speed, also leads to a decline in the quality of the generated RNA data, especially in the generation of details and complex structures, which is not as good as the traditional diffusion model. At the same time, its generation speed is still unsatisfactory, and its acceleration effect is not obvious when generating large-scale data. In summary, current single-cell RNA sequencing data generation methods face three major bottlenecks. First, the generated data lacks biological fidelity and consistency. Synthetic data often fails to accurately capture the complex gene expression distributions, particularly in highly heterogeneous or rare cell populations. Second, precise control over generating specific cell subpopulations remains challenging. Most existing methods cannot perform conditional generation within a unified framework, instead requiring separately trained classifiers or filtering strategies. Finally, generation speed is prohibitively slow with high computational costs. So, to address these challenges, we propose ESCFD, one efficient single-cell RNA sequencing flow diffusion model. Our method maps discrete single-cell RNA sequencing data to continuous representations using an autoencoder, optimizing diffusion model training and improving computational efficiency. During training, we designed a pre-guidance mechanism that utilizes cell representations learned from pre-trained self-supervised models as guidance information, aligned with diffusion model representations through simple regularization to enhance the quality of generated samples. In the sampling process, we adopt the concept of probabilistic flow odes and design a post-guidance mechanism, using an efficient ode solver to significantly reduce time costs. Additionally, by introducing label information through a classifier-free guidance mechanism, our method can generate different types of cells without requiring an additional classifier. As we know, the single-cell sequencing data is usually bag-of-words data because there is no upper limit on the gene expression value of each cell, and the order between genes does not affect the cell diffusion models that specialize in processing continuous data are not suitable for processing discrete bag-of-words data. The latent representation generation model with an autoencoder can map single-cell RNA sequencing samples to a continuous latent space, allowing the diffusion model to be trained. In addition, it can access an efficient low-dimensional latent space where high-frequency, imperceptible details are abstracted away. Compared with the original data space, the latent space is more suitable for the use of our method in this task, because it can now be trained with continuous data, focusing on important gene information in the data, and trained in a more computationally efficient low-dimensional space. We map the original data to the latent space through a pre-trained autoencoder round and map the generated data to the original space through a sub-encoder map. While generating realistic single-cell RNA sequencing data is highly challenging because it needs to capture complex and nonlinear gene expression patterns while accurately modeling structural differences between cell states in high-dimensional space. We noticed that in the field of computer vision, the REPA method achieved remarkable results by introducing a self-supervised visual encoder to extract high-dimensional structural features of images and learning to generate more realistic images through the diffusion process. Inspired by this, we considered whether the biologically meaningful expression information learned by self-supervised encoders could guide diffusion models to learn higher-quality latent representations. Therefore, we proposed a pre-guidance mechanism that combines a pre-trained self-supervised encoder with a diffusion model, leveraging the former's capability in modeling gene structure representations. The implementation of this mechanism is concise and effective. We first use the self-supervised encoder to extract the external representation ZS of the input cell, then extract its internal latent representation ZM from the intermediate states of the diffusion model's transformer. Next, we perform regularized alignment between the two through a simple alignment loss, thereby forcing the diffusion model to continuously focus on gene representation information during generation. This soft guidance approach not only avoids reliance on discriminators but also significantly reduces training instability. Although the traditional diffusion model has superior generation quality, its sampling process is extremely inefficient, and it usually takes thousands of steps of step-by-step denoising to complete the generation of a sample. This high computational complexity is particularly prominent when processing single-cell RNA sequencing data. Because single-cell RNA sequencing data itself has the characteristics of high dimensionality, high sparsity, and large sample size. This makes it difficult to promote the diffusion model to practical applications on a large scale, especially in large-scale generation tasks that require simulation, disease modeling, or drug screening, where efficiency becomes a decisive bottleneck. So we designed a post-guidance mechanism in our method. Its core idea is to transform the original random diffusion sampling process into a smooth, deterministic probability flow process. We guide the sampling path to evolve towards the target distribution by modeling the velocity field. This reconstruction perspective no longer relies on the random process of step-by-step sampling, but uses an efficient ODE solver to directly solve the path, greatly reducing the number of sampling steps to a few tens of the original while maintaining the generation quality. In addition, we integrated the classifier-free guidance mechanism. During the training process, we introduced cell category labels as conditional signals for the model and trained an unconditional branch without labels. In this way, during inference, no external classifier is required, and flexible control from random generation to directed generation can be achieved by simply adjusting the guidance strength parameter lambda. This not only improves the generation stability and controllability of the model, but also greatly simplifies the system architecture. In experiment part, we conducted systematic tests on 3 mainstream single-cell RNA sequencing benchmark datasets, including 68,000 PBMC, tabula muris, and brain small, covering complex cell populations from the human immune system, multiple mouse tissues, and the cerebellum, respectively. For evaluating the quality of synthetic data, we adopted two strategies: qualitative visualization and quantitative metric analysis. First, for qualitative evaluation, we used the U-map dimensionality reduction tool to map real and generated cells into a low-dimensional space to visually compare their distribution overlap in the cell lineage space. Next, for quantitative evaluation, we used two core metrics: AUC, it's the area under curve, and MMD, it is the maximum mean discrepancy, which measures the overall difference in expression distributions between generated and real data, with lower MMD indicating higher similarity. As shown in this figure, we generated cells for PBMC and muris, both overall and specific subpopulations, nearly perfectly replicate the real data's distribution, preserving both the overall structure and marginal rare populations. By combining qualitative and quantitative evaluation methods, we effectively assess ESCFD's performance in generating synthetic RNA-seq data highly similar to real biological samples. Table 2 lists the evaluation results of deep learning-based methods and ESCFD, showing that ESCFD significantly outperforms other methods. Additionally, we conducted ablation experiments on the pre-guidance mechanism. We further conducted downstream analysis on the consistency of gene expression levels in the generated samples. First, we selected several key genes and plotted QQ-plot curves on the PBMC and tabula muris datasets. As observed in Figure 5, all curves are close to the diagonal line, indicating that the model maintains a high level of consistency at the gene expression level. Next, as shown in Figure 4, we verified the conditional generation capability and controllability of our method. We conducted multiple lambda-guided experiments by adjusting the lambda value to control the model's generation willingness strength. In the visualization results, when lambda is set to 1, the model operates in an unconditional generation state. As lambda increases, the generation becomes more biased, explicitly shifting toward a specific cell type. Finally, we evaluated the advantage of our method in terms of generation efficiency. To validate whether the post-guidance mechanism improves sampling speed, we compared the time required by 4 models to generate 3,072 cells under the same hardware and batch size. The results show that it only takes 37.77 seconds, which is 3.73 times faster than the current fastest model, like Diffusion. This result confirms the significant efficiency advantage of our proposed post-guidance mechanism. Okay, this is the details of our work, and thanks for your listening. In future, we will try to optimize the training efficiency of our method to further save the cost required to train RNA-generated models. And if you have any questions, please send emails to us, because I cannot answer them in person right now. Thank you for listening again. Appreciate it.